So, very good afternoon to everybody. It's fortunate that I am speaking after Amit and other previous speakers, which is they have introduced many of the targets, the complexity of the targets for genome editing. Today, my presentation, I will not go elaborate into the CRISPR aspects. I will be only limiting my talk to whatever is applicable to phytoplasma in a generic fashion. So many of you may be aware about CRISPR-Cas, like the genome editing, which is mediated by different Cas enzymes. It's based on the CRISPR. Originally, which was identified in Streptococcus pyogenes, but now we have varieties of Cas9. Okay, so this is nothing but it's not invention. It is already existing immunity based reaction. This is more evident in bacteria and archaea. So this is based on the adaptive immunity of bacteria based on the nucleotide memory of the previously invaded phages. So this is very similar to RNAi, which is emanated from the basal eukaryotes as against what we see in the prokaryote. So CRISPR-Cas9. So CRISPR-Cas9, the knowledge of the CRISPR-Cas9 started way back in 1987. It's not that the Nobel Prize winning study, Jennifer Doudna and Charpentier in this year was actually started by a PhD student in 1987 in Spain. So that small observations at the microbial repetitive elements, so that nobody knew that it's going to develop into a so profound technology like following RNAi. Okay, so there were questions about RNAi, so I will not answer RNAi at this point of time because we are concentrating more on genome editing. So all this started with the phage infecting the bacteria and the bacterial response or the immune response to the phage infection. So the 30 years of knowledge, it was revolving more around bacteria or more around microbiological investigations. So no eukaryotic scientists were much interested in knowing what is the immunity of bacterial immunity towards phages. So if you look at the contemporary genome editing technologies, so it is a convergence of two research frontiers. Okay, so it is not that it is existing in eukaryote. So it is essentially emanate from the DNA, exploiting the existing DNA repair mechanisms in the eukaryote and the CRISPR-mediated immune system in the prokaryote. Okay, so it is a convergence or the marriage of these two ideas. If you look at the native bacterial immunity, so we need to understand the both sides of the coin. So in case of native bacterial immunity, we have when the phage attacks the bacteria, so the bacteria defend the incoming phages in 3 steps. One is by acquisition, whereby a portion of the phage DNA is incorporated into a locus called CRISPR. Then there is a biogenesis of CRISPR, pre-CRISPR RNA, that gets associated with the trans-activating RNA with the Cas9 protein. So that is going to interfere with the phage, and then it is going to get the phage DNA gets cleaved. So it is going to kill the invading phages. So this genetic memory is passed on to the daughter cells. So we know that E. coli replicates every 20 minutes. So when the phage, the daughter phage, if it is encountered, if it is encountering the phage, the same phage attack the next time. So it is the genetic memory kicks in, and then the CRISPR RNA and the trans-activating CRISPR RNA associate themselves with the Cas9, and it is going to kill. Okay, so that's how the propagation of the bacterial immunity is happening. So if we understand, we have 3 components. One is called the CRISPR RNA. Then we have a trans-activating CRISPR RNA. Then the Cas9 protein. So the Cas9 protein, as you can see, so these are all in the upstream of the CRISPR locus. Okay, so these are all Cas-associated genes. So we have n number of Cas-associated genes now with the different systems. And with the recent classification of the CRISPR systems, we have mainly type 1 and type 2, and we have the several classes. So the several classes have different functionalities for which we can use for editing either insects or the plants, whatever it is. So we have the capacity to edit any locus let it be a heterochromatin or euchromatin area. So any locus, any place, so it is possible to edit. So edit, well, I would come to the slides like what is editing. Okay. So the natural, so what we need to understand is the native bacterial immunity. We have three components. One is the CRISPR RNA, which has the similarity or the complementarity to the incoming phage DNA, and the trans-activating RNA, which has a scaffold, which act as a scaffold. That means a physical binding to the Cas9. So then the Cas9 looks for a signature called PAM, that is called protospacer adjacent motif. So that is a signature. Following the PAM signature, the Cas9 protein binds to the target with the CRISPR RNA, or which is having 100% complementary to the incoming phage. Okay. So the other aspect of the eukaryotic system is when there is the eukaryotic DNA can be mutated in different ways. There are physical forces, ionic forces, X-ray. So there are so many ways. The current technology of genome editing has the convergence of two research frontiers. Both are naturally occurring. It's only it's a very intelligent marriage of two ideas mediated by Emmanuel Charpentier from France, as well as a very famous microbiologist, and also by Jennifer Doudna at UC Berkeley. So they're just a very curious idea of making this sgRNA by joining CRISPR RNA and trans-activating RNA. It has opened a profound area of medical therapeutics as well as for agricultural applications. So the first part of the genome editing emanated from the understanding the native bacterial immunity. So in bacteria, they defend the incoming phages by taking a piece of their DNA or RNA or any plasmids which are infecting in the bacterial cell, and it is incorporated into the locus called CRISPR locus. So in CRISPR locus, upstream of the CRISPR locus, we have the genes, the protein genes which are associated. These are called Cas genes, that is CRISPR associated genes. So then again, upstream to it, it is a trans-activating RNA, that is a tracrRNA. So there are three components in the native bacterial immunity. One is the CRISPR RNA, which has the complementary sequence of the invading phages, and the trans-activating RNA, which interacts with the Cas protein and act as a scaffold, or it is a physical support to the CRISPR RNA to act. Okay, so when the daughter cell has the incoming nucleotide matches with the CRISPR memory, then it is going to cleave by three steps. That is by biogenesis, then like adaptation, biogenesis, and interference. So the last step is similar to RNAi. So there is a little bit of dissimilarity. So the other part of the story is the double strand break or the single stranded break or SNP mutations which are occurring in the eukaryotic cells, which how they are getting repaired. So usually there are different agents that causes the natural break. Okay, that by physical shear or by irradiation or by ionic radiations or cosmic radiations or by X-ray. So the double eukaryotic cell repairs the mutated gene or the mutated DNA in different ways. One is if they are having double stranded break, it indicates the two pathways called NHEJ, that is non-homologous enjoining, and we have another type called homology dependent repair. So in case of NHEJ, next slide please. Okay, so we will not bother about like other ways the DNA getting repaired. We will bother only about the double stranded break, so which happens in the prokaryotic community. Okay, so in case of double stranded break in the eukaryotic cell, this is immediately repaired by there are hosts of proteins which are involved in this is called based on the Q proteins, and finally what we get is so we have religated DNA, but the efficiency of religation is very poor, and then it introduces indels, that is insertion deletions. So in a way, it is mutagenic. That means this error-prone repair mechanism, repairing the double-stranded break, is inefficient in terms of the native DNA repair mechanisms. But we take advantage of this error-prone repair for editing. So previously, this CRISPR-Cas9, as I was telling you, it was more confined only to the prokaryotic defense, and it was for 30, 35 years, it was only with the microbiologist, and they were trying to understand what are the different, how different bacteria, both gram positive and gram negative, they have the different systems, different systems based on the CRISPR, and what are the different enzymes or the different Cas9 proteins are associated in degrading the incoming phages or the plasmid. Okay. But when it is changed into a repurposed editing of eukaryotes, that opened the way, as I told you, into a very interesting field of medical therapeutics, as well as like improving crops, even without any DNA. Like, for example, without having to develop a transgenics. Okay. So now the transgenics, the development of transgenics is not so favored in our country. So far, there are, except cotton, we don't have any transgenics, and there are many transgenics are in pipeline, but they are not released. But this is a CRISPR-Cas9 has made us to develop any crop or edit any organism in a non-transgenic way. That is a DNA-free mechanism. So this is naturally equated to the natural mutations which are accumulating in organisms, so that there is a less deregulation process associated with any development that we do with the CRISPR-Cas9. So recently, DBT has come up with a guideline. They grouped into this genome editing into 3 groups. One is SDN1, 2, 3, depends on what type of editing we do. So if you are simply making a double-stranded break of an organism, and the natural ligation takes place, that falls into SDN1 level. And if you are mutating a single pair, a base pair in a gene, that falls into the SDN2. And if you are using homology-based repair, that's HTR, so that falls into SDN3. So essentially what we have to capture is SDN1. And the initial draft guideline says that any organism which is developed based on SDN1 can be released through your institutional IBAC. So that is the big leverage given to for a releasing. Otherwise, the very lengthy process of deregulation of any transgenics, including RNAi. So this is a wonderful, very small input, but it is a very excellent input by Jennifer Doudna as well as Charpentier. They only combined this spacer, that is a CRISPR RNA and the gRNA. Okay, so this is a trans activating RNA. So just by fusing this, they made this more applicable to eukaryotes. Otherwise, eukaryotes, we do not have any CRISPR-based defense, and this has made a very good input into editing eukaryotes. So we have now varieties of Cas9, Cas proteins, I would say, and this gRNA plus spacer, this scaffold plus spacer is called single guide RNA. That is called sgRNA or guide RNA. So this this complex is known as RNP, that is RNA and Cas9 complex. So using this complex, RNP complex, so this favors DNA-free editing. That means this can be equated to a natural mutations that breeders when they do the irradiation. So okay, so we can also develop transgenics. For example, I would come into the last part of my presentations, whether we for phytoplasma, whether we need to develop a transgenic approach or we need to have a DNA-free editing. So just a graphical representation of this target DNA. So as I told you, this is a very important, 2 signatures are important. One is the Cas9, this is a protein, yellow one is a protein. And this looks for a signature called PAM, that is the trinucleotide for SP-Cas9 only, that is NGG, that is any nucleotide and GG followed by GG. So, and following this signature, identifying the PAM, the CRISPR RNA is going to have a complementary base pairing to the target. Okay, so as I told you, this sgRNA, that is single guide RNA, this is what we see. This loop, the tetra loop, is known as trans-activating RNA. So now this is fused artificially, and this has become a single guide RNA. Okay, so once the target is recognized, so it is going to make a double-stranded blunt and cut three to four nucleotides upstream of the PAM. Okay, so we know that when the double-stranded break happens in the eukaryotes, it is going to be naturally. So it is going to be repaired two mechanisms. One is, as I told you, NHEJ, which happens at 99.9% of the time. That is non-homologous engineering. The other method, if you have if you supply the correct copy, then it is going to undergo HDR, that is homology dependent repair. So we are not going to bother really about the homology dependent repair. In case of agriculture, so we are entirely depending on NHEJ method. And very recently, Jennifer has also developed a very interesting Cas9 protein from the phages itself. We will see at the end of the slide. And also, there is a very important input from David Loof lab in the MIT Broad Institute. So they have made another improvement in removing some of the shortcomings of the double-stranded break using primary TET3. So what we need to understand is we are going to cleave any GNA or any RNA. So again, depends on what kind of Cas we use. So if you use a Cas9, it's going to target RNA-guided DNA cleavage. So it is going to result in double-stranded break, blunt and double-stranded break. So this break will be repaired mainly by NHEJ. Since NHEJ is error-prone, so that we are going to get that particular gene mutated. That is, if we are able to get in case of frame shift. So if it's not that if we are going to release our large chunk of DNA for us to have a mutation. So it can be a single nucleotide removal or insertion, but finally what we need to have a frame shift. So if there is a frame shift, the entire protein folding is going to be changing. So the active sites are not going to be sitting in the proper conformation, so it is not going to be functional. Okay, so this is the advantage using double-stranded break. Now we have other advancements in not having double-stranded break, but we have a staggered nick in the DNA.